Okay, good day everyone. My name is Sarah Adana Ene, and I'm a graduate student here in Dr. Banerjee Lab here at Texas Tech University. So I am very interested in researching on diabetes, metabolism, nutrition, epigenetics. These are areas that I'm working on or I'm hoping to work on in the nearest future. Now, diabetes is actually a metabolic dysregulation that is plaguing many people around the world, millions of people around the world, and has impacted a lot of economies. It is a menace that has needs to be researched on in order to come up with a lasting cure for this menace. Now, that while therapeutics have done a lot to manage the situation, it has not really done a lot in reversing the diabetic conditions in patients. And this is why I have vested interest in doing research in this area. So before now, I had done research on the impact of herbal remedies, that is uh, Ocimum gratissimum and Verononia amygdalina, on glycated hemoglobin species. Now, glycated hemoglobin is actually a marker that clinicians use to diagnose the progress of diabetes, especially on a 3 month basis. Now, my findings, was that these herbs, glycated Veronia amygdalina and Ocimum gratissimum, were actually able to lower glycated hemoglobin species and hence had the potential to reverse the exacerbation of the disease or the progression of the condition. And so, but that research had some pitfalls. They had some questions that were unanswered. Like, for instance, by what mechanisms were these herbs able to mediate this glycated hemoglobin lowering ability? Was it by impacting molecularly on the hemoglobin, or even in influencing the metabolic process of glucose? I couldn't answer those questions. And so to answer those questions, I'm here at Texas Tech studying microbiology, particularly the cell and molecular biology part, where I'm deploying certain cell and molecular biology techniques such as quantitative PCR assay to be able to quantify some of these regulatory and transcription genes that are able to regulate these pathways that are involved in energy expenditure and metabolism. I'm also employing in situ hybridization to be able to detect in vitro transcription of genes in particular tissues, specific tissues in the body that are involved also in metabolism. I'm also deploying other solar molecular biology techniques such as transfections, cytotoxicity assays, Bradford assays, Western blot assays, immunofluorescence assays, and I'm looking forward to doing also reactive oxygen species ATP assays to give us an idea of what is happening in the mitochondria, because of course diabetes results when there's an imbalance between energy intake and energy expenditure. So by the time we do the reactive oxygen species as well as ATP assay to give us an idea of how these regulatory genes in metabolic pathways are able to impact ROS generation as well as ATP. I also want to investigate the impact of these regulatory genes that I'm very concerned about on even metabolic pathways such as glycolysis, gluconeogenesis, glycogenolysis, and the lipogenetic pathways. So I want to compare all this with maybe controls like inhibitors and so on and so forth. So I actually I have a background in human physiology, where we study human you know human mechanism of action in different systems of the body. We're able to relate that with pathophysiologies and be able to project what possible exogenous agents can mimic such events in the body, And when I mean exogenous agents, I'm not just talking about therapeutics or drugs. I'm also relating it with nutrition. Some of these necessary things that we may interact with our body when we take them, and how they impact on our genes and even influence disease outcomes in patients. So these are areas that I'm actually interested in and I'm actually working on, and I'm open to collaborate with partners. I'm open to collaborate with research institutions. I have been doing some research with medical research institutes internationally, and I am also looking forward to um, such partnership globally, um, to do not just metabolism but also nutrition and epigenetically, to study how we can come up with some healthy lifestyle or nutrients that we can take that will influence some nucleotide polymorphisms that are healthy and that can in the long run reverse diabetic phenotypes in diabetic patients. So I really thank you so much for listening to my video this day, and I'm hoping to come up with more updates as my research unfolds, and I also look forward to feedback, contributions, partnership, as the case may be. Thank you and God bless.